Hello, everyone. This is my draft presentation on my project looking at glacial influence on carbon cycling and freshwater microbial communities in the Canadian Rockies. So first off, glaciers have recently been demonstrated to be key players in the carbon cycle. And they can do this through storage of carbon through atmospheric deposits on the surface, overridden vegetation at the surface bed and in between, and through microbial metabolism that happens at the surface and the base. So microbes eating plants, each other in the atmospheric deposits. So all this carbon gets stored within the ice, and then when the ice melts in the summer, it gets exported into downstream um, ecosystems. And where this is a alpine glacial system, it is getting exported into freshwater. There are also other components that get mixed in with these glacially fed streams, such as terrestrial inputs from vegetation. So what's important to know is that when these carbon sources mix, they're actually still quite different. So glacial carbon and terrestrial carbon are not alike. Glacial carbon has low concentrations of carbon. It's very old and has microbial-like features, such as being 13C enriched, having low humic components and low aromaticity. Whereas terrestrial carbon has high concentrations of carbon. It is young and it has plant-like features, such as being 13C depleted, having high humic components and higher aromaticity. So all of this to say is that glacial carbon is representing a unique carbon source to freshwater ecosystems compared to non-glacially fed rivers. So within streams, organic carbon is actually very important to heterotrophic bacteria. So it gets assimilated by heterotrophic bacteria, and then it can be passed on to higher trophic levels that are eating these bacteria. And alternatively, they can respire it as carbon dioxide into the atmosphere, which means that dissolved organic material is leaving the system. They can also break down larger pieces of organic materials, particulate organic material, breaking it up into dissolved organic material, which can then either go again into food webs or exit the system as CO2. And evidence has found that glacial carbon is actually really important to this system in glacially fed streams. So it is preferentially being taken up by heterotrophic bacteria over plants. So you have an accumulation of carbon sources in food webs and being respired rather than other components in the system, such as from plants. So the objective of my project was to investigate how carbon and microbial community composition is characterized in the Canadian Rockies, because we have little data in this region. And the second was to explore how glaciers are influencing carbon characteristics and microbial community structure. And the third was to explore how variations in the carbon pool are driving variations in microbial community composition. And this is all looking at the question of helping to understand, when we lose glaciers in this region, what will happen to this ecosystem? So how important are these glaciers to carbon pools and to microbial communities? So to do this, what I did was I sampled 4 glacially fed rivers in Banff and Jasper National Parks, and here are just the maps of our sampling locations, and for a total of 12 different sites. I sampled them monthly from May to October, and the reason that I did this is because there are difference in stream water contributions throughout a season, with spring representing primarily snowmelt and late summer being glacial melt. So the idea behind this is that if there are differences in season, potentially it could be due to glaciers, as glaciers have strong influence in the summer, late summer. Additionally, these samples were taken along a range of 0 to 100 kilometers from glacial terminus. This here is a picture of the Athabasca Glacier headwaters, and this is a picture of the NSR River about 60 kilometers downstream from the terminus. And as you can see, they look quite different. So as we move along this gradient away from the glacier, we get increasing terrestrial inputs into the stream and decreasing glacial inputs. So what we'd expect if glaciers are structuring carbon pools and microbial communities, that we would get a difference from a site close to a glacier versus a site relatively far away. So the parameters that I looked at were dissolved organic carbon, absorbance characteristics such as A254, SUVA, and S275, S295, which can be used to infer things such as humic components, aromaticity, and molecular weight of the components. Additionally, I took 14 C samples, which can be used to determine relative carbon age. And I took 13C samples, which can be used to look at sources of carbon, whether plant or microbial-like. And finally, I took samples to be used for microbial community analysis. So looking at some exploratory analysis that I have done so far, I have focused mainly on objective 2 on how glaciers are influencing organic carbon or microbial pools. This here is a PCA that I did looking at some of my organic carbon parameters that I collected. And I colored them by month. And what stands out to me here is that the samples from May are separating out from the samples taken at other times in the season. And they're separating out because they have high DOC, high A254, and high SUVA, which is indicating that they are more plant-like and have maybe more terrestrial components than those taken in the rest of the season, which look more typical to glacial sources. What also stands out to me here is that distance is inversely related to S275 to 95. So as we move away from the glacier, we are actually seeing a decrease in the molecular weight of our carbon pool compared to close to the glacier where we have larger molecules present. Additionally, I added different shapes to represent my different rivers, and it doesn't appear that there's any separation due to river type, meaning that these trends are similar in all the 4 rivers that I sampled. Next, I looked at %13C and %14C at my sites, and I separated these from my PCA because logistically, I could only get a few sites for these analyses. What I noticed most about %13C here is that it's within the range of -28 to -16, which is actually similar to a plant material, which shows me or suggests to me that perhaps this is evidence for plant material in the organic carbon pool that is not really different over distance or month. And even though they're showing distance, it's still within this terrestrial range. Next, looking at %14C, this is actually indicating a pretty old carbon pool, and it is actually increasing in age as we move further away from glaciers, which is what I would expect to see. And it's also maybe suggesting that this old carbon is being actually used up as it moves down the river. So perhaps it is being preferentially used over terrestrial sources as we move down the river. Additionally, I did box plots of these by month, and the lowest values of my %14C that I obtained were actually from the month of August, which I think is interesting because that is the month with the highest glacial melt rates. Looking next at my microbial samples, I did a principal coordinate analysis of my microbial species that were present in my different samples. And I colored them by river, month, and by distance range. And my key takeaways from these analyses were that it does appear that there is a difference in season to see what microbial communities you'll get. So microbial communities that were taken in May are quite different than microbial communities found later in the season. However, there is not much separation based out on river or by distance range, meaning that potentially these are not important factors in structuring microbial communities. Additionally, I did an indicator species analysis, which is just showing here that, which I colored by month, which is just showing here that samples that were taken in May have species present that are indicative of May that are actually not present in the rest of the samples or present in lower amounts than they are in the May samples. So this is just more evidence that community is structured different in May than it is to the other months. So the next steps for my analysis are to investigate and to compare other potential end members such as snow and groundwater sources to better understand the influence of glaciers seasonally. And secondly, to explore if variations in organic carbon are related to differences that we see in microbial community composition. And the main strategy that I'm going to take to look at this is a redundancy analysis. Thank you for watching my presentation.